Right, so good afternoon and welcome back to our conference, to the final session of our conference today. And my name is Jutarop, you can call me Peach, it's my nickname. So just a little bit about myself. So I now I'm in charge of the director of the Kaen University Phenome Center. And as a disclaimer, I have been staying very far away from cancer immunotherapy at first. But I would like to thank Professor Nattiya and also Assistant Professor Supanika and the TCCI team for bringing me into this world of cancer immunotherapy. And I hopefully find myself useful in this hub. And let's have a look at what I've been sharing today. Right. And this is the overview of my talk today. So I will start with the concept of metabolomics, followed by the applications of metabolomics with the metabolic characterization of cell and cell constructs. And I will share with you a very little piece of work that we have been just starting our collaboration between Chulalongkorn University and ourselves, Kaen University, on the metabolic characterization of different cell constructs and proficiency tests for potential multi-center analytic units. Let's begin then. So, if we talk about metabolomics, it can't be denied that we need to start with this very ancient textbook. So, does anyone know what this textbook is all about? Or has anyone read it before? It looks so ancient, doesn't it? So, this is called Corpus Hippocraticum. So, it's actually a compilation of texts recorded by Hippocrates and his colleagues and students during the classical period. And at one of the sections, or one of the sections in this textbook, it mentions about prognostics. So when we talk about prognostics, it's something that actually not new. So it has been established, since classical period as such. So dated back so many hundred years ago. So doctors during that time, they were not allowed to ask the patients to disrobe themselves. So that means the patients need to come with, a jar of a urine. This is the furthest sort of approach that they can, help with the diagnosis of the doctors. Apart from looking at the signs and symptoms that they can, look on the patients. So this is just the old painting, from some of the wall in the cave in the past years, and you can see that patients came in with a jar of the urine, and then doctors during those days, they need to observe the characteristics of the urine, and also they need to smell the urine, and of course they need to taste the urine. So why do they have to test the urine? The reason for that is because they would like to get as much information as they can in order to construct this kind of pie diagram. It's called the urine wheel during the medieval time. So this is the history of metabolomics. So metabolomics itself is not new. So the concept of metabolomics was established since the classical period. So in this urine wheel, it would indicate the color and as the appearance, the texture of the urine, the smell, and also the taste of the urine, that in accordance to the signs and symptoms that the doctors observe from each patient. Then this is some kind of like a Bible book, for doctors during those days. But luckily, after the science revolution and also industrial revolution in England, so technologies evolved and developed. So on the left-hand side is the metabolomic timeline from 1946 to now, but just a brief, summary of this timeline. You can look on the right-hand side panel, which I summarize here. So in 1940, Roger Williams suggested that each individual may have a metabolic profile. So that means during that time, they believed that each and every one of us would have or possess the distinct and our own metabolic profile. Then in 1971, GCMS was first used to measure compounds in human urine and tissue extracts, and the term metabolic profile was introduced by Honing in that year. Later in 1998, Professor Jeremy Nicholson slightly turned this term from metabolomics to metabonomics. Have you heard of it? So if you read some literatures or articles, you will find a variety of the terms we used for describing metabolic profiling. We can use metabolomics, metabonomics, metabolic phenotyping, metabolic profiling. So all of these share the same sort of definitions, but all of them would have somehow the unique definition or characteristic to describe each one. For metabolomics, the L, and metabonomics, the N, has a slight difference between the two. So for metabolomics is when we look at the metabolic profiles of such living organisms or in the biological system as they are. So without intervention, without treatments, without responses. But when we look at the responses of some living organism or biological system, in regards with some stimuli, in regards with some drug treatments, diet intervention, we rather use the term metabonomics with the N. So this is to distinguish between the two subset of the studies to make it clear from the beginning. So in 2005, there was the first metabolomics web database called MedLin that was developed. And in 2007, people started to have a first draft of the human metabolome, and that was later completed in 2012. And now it is time for applications in utilizing metabolomics. So metabolomics, if we use that or apply metabolomics approach in clinical study, we call clinical metabolomics. So just a bit of the introduction, why metabolomics? So we know genome for years, like many, more than 2 decades, I would say, after the Human Genome Project was completed. And genome tells us what can happen, while transcriptome tells us what appears to be happening. So we are getting closer to the cause of, the phenomenon that we are observing. And then proteome tells us what makes things happen. So proteome data would tell us basically the protein domains that drive such metabolism to take place. And for metabolome, it tells us what is happening right here, right now. So that means, well, I can't really tell which omic is more important than the others, but all of these omic data or layers, need to be integrated and complemented in order to understand, the etiology of diseases, in order to understand the responses in mechanisms, in molecular mechanism point of view, and also to understand the interaction between different biological signals living, residing in our body or in the plants or in animals or in bacteria as such. So as you can see from this diagram here, metabolome is actually closest to the phenotype. So that means for the downstream utilization of these biological signals, metabolites can be very easily applied into the real use. For example, the detection of metabolites, which are much more straightforward than detection of genes and much cheaper than the gene sequence. And when we talk about clinical metabolomics, what are these metabolites that we are looking into? So the metabolites in clinical metabolomics include amino acids, nucleotides, steroids, free fatty acids, neurotransmitters, bile acids, creatine-related metabolites, or some lysozyme, peroxisome metabolites, this kind of thing. So these are the metabolites that we will look into clinical metabolomics, or any other di or tripeptides that have the molecular weight of less than 1,500 Daltons. So now let's have a look into metabolic phenotyping. So for metabolic phenotyping, why metabolic phenotyping is so important nowadays? This is because the phenotypes that have been dynamically changed over time or over seconds, it can be a result from the genetic and epigenetic factors, age, environment, diseases, or some drugs, or some lifestyle types like smoking or non-smoking, nutrition, and all of these factors contribute to the changes or the transient and quick alteration of the metabolites in both concentration and types. So metabolites changes can reflect into two arms. One is for health monitoring that can be used to help track the changes of the phenotypes, and also is for risk prediction as well. In order to construct a metabolomic study, so metabolomic is as I mentioned, a systematic study of all metabolites quantitatively and qualitatively that are present in a biological system. So the very first step actually is not listed on the pipeline here on the screen. Actually, it is the study design. So this is the most critical step in metabolomics, not only metabolomics, but also in other omics studies like genomics, transcriptomics, and proteomics as well. So if we have a very poor study design, we're going to end up with a very poor sample collection, poor data acquisition, very poor statistical analysis, and also the outputs, So it's just like rubbish in and rubbish out. So we need to very carefully plan, our study before start taking the samples from the site of study. Then for biological samples, we need to think about what could affect or what could act as the confounding factors in such studies. This is because metabolomics, and other omics, but especially this one, is very highly sensitive. So any small intervention or stimulus, can change the alteration of the metabolites that you detect. So that means we need to be really careful in terms of sampling and also the tissue and biofluid collection, because we have to think about the confounders at all times just to prevent ourselves from introducing the confounders into our study. Then, in terms of the instrument of choice, we have not many options. We have NMR, nuclear magnetic resonance spectroscopy, or mass spectrometer, either coupled with gas chromatography or liquid chromatography as such. So we have got about, let's say, 3 major platforms, but some people, they try to develop, the metabolomic study using, for example, Raman spectroscopy, but Raman has a much lower sensitivity in comparison to MS, So, but it would be more practical for Raman spectroscopy because it has a portable version that people can bring into the site and get the sample and measure some certain metabolites straight away. That kind of example. But nowadays, for research and development, especially for discovery phase and validation phase of the metabolic biomarkers, we rather use either NMR or MS only. Then, in terms of data pre-processing, the statistical analysis, we need to highly utilize bioinformatics. So when I talk about bioinformatics, it's not just how we process or handle the nucleotide data, but this time, bioinformatics is mentioned in the context of the metabolites and or proteins. So that means these require different skill sets, of the researchers or the analysts in order to analyze a different data sets. In this case, it's metabolite data. So we need to be able to pick up, a very sensible and logical statistical pipeline as well as the bioinformatic pipeline or tools that can be applied for such data sets. And then I would say the most challenging step in metabolomic workflow is this one, is the compound identification. Does anyone know how many metabolites do we humans have? Any guess? 10,000, okay. Who gives higher or lower? It's not bidding, by the way. I don't have anything for you to give away. So for
5,000.
5,000 from where? From here? Okay, 5,000. So the known metabolites that humans have is about 250,000 metabolites. 150,000. 250,000 metabolites that have been identified. But there would be some other transiently changed metabolites or transiently formed metabolites that our technology nowadays cannot be used for detection. So that means there will be 250,000 plus that we don't know yet. Okay? So, but for those 250,000 that we know is already difficult enough, right, to identify all of these metabolites in our data sets. And especially for NMR or nuclear magnetic resonance spectroscopy. So, so far, there is one company trying to develop the AI, model in order to automatically identify the metabolites, but they fail big time. So because some metabolites, like if you look at the metabolites, they are chemical structures, right? And for example, arginine and argininosuccinate, these two metabolites have very similar in structure. So sometimes the NMR resonance coming up at the same, chemical shift. So the software itself cannot distinguish between the two. So it still requires, manpower, like manual manpower, in order to identify one by one. And I would say, well, Dr. Mahidol is one of the best persons to consult with this. And last but not least is the biological interpretation. Because metabolome data is not that, it's not straightforward. it's not like, if we know the gene, right? We know what this gene expressed into which kind of expression, right? Or into which kind of mRNA, for example. But the metabolite, one metabolite can be involved in more than one pathway. So we don't really know, what's going on at the time that we collected the samples. So metabolomics is actually a snapshot of the metabolic photograph, that by the time that we collected the samples. So that means it could be one or the others. So we have to have, some other omic data in order to help interpret the metabolome data in this case. So having talked about metabolomics, so this slide shows the applications of metabolomics. So it can be used in so many fields of research and also in so many aspects of industries. So it can be used in medical sciences, like for example, the biomarkers discovery of cancer, or it could be used to track the stages of the kidney and urological disorders. It could be used to characterize the metabolic alteration or signatures in inflammation in immunology, and it can be used in animal health, in agriculture, in mechanism of action of drugs or novel drugs. It can be used in microbiome studies as well, because most of the microbiome metabolites can be detected using metabolomic platforms. So that means if we finish the sequencing experiment and we know what sort of microbiota are there or reside in the system, and we have done also the metagenomics, that we know the genes that have been involved in such organism or environment, then metabolomic will help complement with the metabolite profile of those microbes, whether these microbes are still functioning by secreting, such active metabolites or not. So for today, we are going to focus on the applications of metabolomics in cancer and inflammation and also immunology. So the disease model that I've used for my talk today is cholangiocarcinoma. So many thanks to Ajahn Usara for introducing cholangiocarcinoma in the previous session. So cholangiocarcinoma is actually a malignant tumor that arises from the dark epithelial cells, and it can be divided into three major types: intrahepatic CCA, perihilar CCA, and ductal CCA. So these are the risk factors, and of course, in Thailand and in Great Mekong sub-region countries, the parasitic infections is a major cause. And this slide shows the global incidence of cholangiocarcinoma worldwide. And as you can see in Thailand, so yeah, this is considered a very high number in incidence. And of course, within Thailand, Khon is a champion still. we don't want to be a champion in this case, but we still consider a champion. And not only that, in Great Mekong sub-region countries like Laos, PDR, some team members of the Cholangiocarcinoma Research Institute, they went out for the field study, and then they collected the urine and tested back at home. And then they found that more than 90% of people residing in three major cities, including Vientiane, Luang Prabang, and Champasak, 90% of the population suffer from liver fluke infections. And this is a picture of liver fluke, Opisthochis vivarini. And this is when they are residing in the dilated bile duct, as you can see here. So I'm not sure if it's moving. Oops. 1 second. Yep, there you go. So you can see that they are here, the transparent living organisms here that are moving, inside the dilated bile duct. And if you would like to have a closer look to these tiny friends, they're here. So this is not all from one person, but this is just actually a leftover from the surgery. Yep. Okay. And for global mortality, of course, Great Mekong sub-region countries and southern China are ranked #1, as you can see from this diagram here. So in our Khon University, we have so many teams working in cholangiocarcinoma and in many fields as well, in cancer prevention, in cancer treatments, in cancer prognostics or diagnostics. So one of the studies that me and Dr. Manida here have been involved is the study of the chemotherapeutic response patterns using metabolomic characterization. So in this study, we would like to identify the predictive biomarkers which can be used to predict the clinical chemotherapeutic drug response of CCA patients. And in order to have such information of the response patterns before conducting the metabolomic studies, we use an assay, the in vitro assay called histodrug response culture, so HDRA, in order to classify, the patients into two groups, non-responders and responders to either gemcitabine and cisplatin or, the other drugs of choice. So then we collected tissue samples, and then we performed the tissue extraction. We conducted the proton NMR experiment on metabolomics, and this is the result. So we found that the response and non-responders to gemcitabine and cisplatin have such a very distinct metabolic patterns in tissue, in the cholangiocarcinoma tissues, as shown on the PSA score plots here. And then we managed to come up with the schematic diagram that underlie the mechanism of what's going on within the tissue, the cancer tissues of the responders and non-responders. So as you can see here, some of the metabolites, for example, some amino acids like tyrosine, histidine, glutamate, valine, methionine are much increased, and also ethanol have been increased in the non-responders. So that means these metabolism of amino acids, including these amino acids as well as ethanol, have driven the resistant mechanism of the cholangiocarcinoma. So at first, we proposed that distinct metabolic signature associated with chemotherapeutic response patterns were evident in this study. However, more precise and personalized chemotherapeutic drug testing platform is needed. That's why one of our colleagues have developed the cell platform for testing the chemotherapeutic drugs. So this study was led by Dr. Pramek Ganrit, Ms. Pimpawadi Pukam, and Ms. Thanaporn Guntawasiri in order to investigate the metabolic baseline characteristic of different CCA cell lines and cell models. When I said cell models here, I meant 2D and 3D, just the monolith 2D and the spheroid. So we constructed the pipeline in order to set up our 2D and 3D cell platforms for testing the chemotherapeutic drugs. And then we for the 3D or the spheroid, we investigated first about the plate, the cell culture plate that we use, either U-bottom or flat bottom, in order to hold the spheroid structure of the cells. And when we found that the U-bottom works much better in order to hold the spheroid shape of the cells. As you can see here, this is a flat bottom that these two cell lines here cannot be formed as a spheroid. So in this study, we use KKU 055, KKU 100, and KKU 213C and 213A. These are the cholangiocarcinoma cell lines in comparison to the cholangiocyte, which is MNK1. And we also perform the Western blotting in order to investigate the cell protein adhesive proteins from either 2D or 3D from each and every cell line. And then we try using different number of cells. So this slide shows the characterization of the 3D multicellular spheroids, and panel A here show the CCA and cholangiocyte cell lines that were varied in seeding numbers from 1,000, 2,500, 5,000, 10,000, 25,000, and 50,000 cells. So we would like to see what could be the smallest number that we could use the cells in order to form the spheroid in this case. And also culture for 48 hours to observe the multicellular spheroids aggregation under the inverted microscope. And we perform the H&E staining in order to see, the segregation and the clustering of the different cells in different zones of the spheroids. And we also do the immunofluorescence staining using DAPI, hypoxia marker, oxidative stress marker, and we merge them up, and we can see that the the 3D multicellular spheroids exhibit typical stonation of hypoxia, oxidative stress, as you can see here, at different density of the cells. Then we extracted all the cells and performed metabolomics using the liquid chromatography mass spectrometer. And as you can see from the very far left PCA score plot over here. So either 2D or 3D doesn't have the strong effect on distinguishing or on differentiating the cells rather than the cell itself, So all the cells cluster together with 2D and 3D, 2D and 3D, and 2D and 3 of each cell line. But then, if we subtracted the mean of the spectral data in order to see the 2D and 3D effects, whether it has any effect on the metabolism of the cells, we can see that the 2 and 3D cell models have such a very distinct and certain metabolic profiles. And then we perform the pairwise comparison using orthogonal partially squared discriminant analysis, which is one of the supervised analysis methods in omics studies. And then, out of a total of 59 outer metabolites that were further analyzed using hierarchical clustering based on the Euclidean distance measure and Walls method for the linkage analysis, the metabolic shifts from 2D and 3D culture system were clearly seen through altering levels of metabolites dependent on the cell types. So this is quite interesting. And as you can see here, the metabolic pathways that underlie the differences between 2D and 3D cells include the alanine, aspartate, and glutamate metabolism, phenylalanine metabolism, and arginine biosynthesis. So these are the top 3 metabolic pathways that are ranked, mostly affected by the 2D and 3D cells. So that means we need to be really aware of, the assays that we would like to test, whether to use either 2D or 3D, because these metabolic pathways have been affected from the very first place. So if you would like to monitor some of the metabolites that are involved in these major 3 pathways that are affected by either 2D or 3D, you need to keep bearing this in mind as well. Otherwise, you're going to get either false positive or false negative data. Then we managed to come up with this schematic diagram that shows the signify metabolic pathways of 2D cell culture and 3D cell culture. For the 2D cell culture, the only metabolic pathway that shows that would differ, in comparison to the 3D, it involves only purine and pyrimidine metabolism, and only AMP, ADP, and UDP that are affected. So that means if any further bioassays that use, these 3 molecules for detection, we need to keep, being aware of this. So otherwise, you may misinterpret your data. And for 3D, as you can see, not only pyrine and pyrimidine metabolism, but other, metabolisms like the energy metabolism involving TCA cycle, glycolytic activity, protein degradation, this kind of thing. So they're all affected. Because in the 3D spheroid structure, it composed of so many subtypes of cells, especially at the necrotic core. So, in this study, we actually investigated the signature of the 3D multicellular spheroids that relies on the bulk analysis. So we haven't done the single cell analysis yet. So we just need to keep bearing this in mind. So that means it is noted that the outer layer of the 3D multicellular spheroids may still be proliferative phenotype, as the cells are still directly in contact with oxygen and nutrients. So that means every single time from now, if anyone would like to use 3D model in order to test the drugs, we need to keep bearing this in mind, not to misinterpret the data. And that leads to this previously published article on immunogenic metabolomics reviews key enzymes that modulate CAR T cell metabolism and function. So this figure here shows the in silico screen and mRNA-seq review the ADA and PDK1 as metabolic reprogramming genes that induce immune response related transcriptomic changes in primary human CD8 plus T cells. So in figure A here, it shows the flow chart of the multi-tier selection for identifying candidate genes that modulate T cell metabolism. And for figure B here, it shows a schematic of the mutant T cell and CAR T cell that can be characterized using the multi-omic integration data. In this case, they use transcriptomics and metabolomics. In this study, it's quite interesting that they use these 2 omics data that they think it would have good enough and more more informative information in order to screen the genes that regulate the T cell metabolism and function. And then they perform metabolomics on the 2 cell constructs, and then they found that different constructs shows just very certain and distinct metabolic pathways involving different metabolic alterations. And if we look at the metabolic reprogramming here, in the ADA and PDK1 mutant primary human CD8 plus T cells, so on the heat map on top of this slide here, it shows the distinct patterns of the metabolic changes in different constructs, between the two constructs, and that can lead to the proliferative and the cell division rate as well. So they match the data of proliferation and cell migration into the metabolic signatures that can help identify the fitness level of different construct of the cells. And in the models, they also integrate the transcriptome and metabolome and to see the highly expressed metabolites and genes that involve in the cell metabolism. Primarily is the energy metabolism and the nucleotide metabolism, because they believe that these two metabolisms would help and involve in the cell proliferation, division, and migration. So the high throughput transcriptomics and metabolomic analysis of these CAR T cells review the alteration of global gene expression and the metabolic signatures in both ADA and PDK1 engineered CAR T cells. And that leads to the new hope in our collaborative research project. So this project has been in collaboration with between Jutarop University and Khon University through the TTCI. And many thanks to Dr. Supanika and Dr. Roslin at Jutarop University to help initiate this study together. And as I mentioned from the very beginning of my talk, in order to construct a metabolomic study, we need to think very carefully about study design, right? I have to say that we have about, I think, 3 times of the meeting that we sat down together either online or on site, and then we come up with our study design here. So we would like to investigate the different cell constructs using metabolomics in order to prove before the actual construction, in order to characterize different signatures of the metabolism of different T cells and also the CAR T cells. And now we just finished growing up the cells and collected the cells according to this plan. And on my end at KKU, we just extracted the metabolites, and hopefully on CU and Dr. Rosalyn just extracted the metabolites as well. So we plan to have things, all the procedures synchronously synchronously connected, at the same timeline, just to prevent them from confounders. And hopefully, we are not only just investigating the biology of the CAR T cells in different constructs, but we would like to do the proficiency testing, between our 2 centers, that we could perform metabolomic experiment in this study in order to transfer the technology between the two. So that means if we come up with the optimized and standardized protocols for characterizing these cells with metabolomics, this technique can be transferred to other universities like PSU, CMU, and Mahidol University as well. So this is our ultimate goal for this study. And hopefully in the next conference, our results would be ready to be presented. Right, so as the take-home messages, so metabolic phenotyping serves as a powerful tool for characterizing metabolites and understanding the fundamental alteration of biological processes following a particular intervention. And this approach is to deal with molecular big data in combination with the metadata and clinical data to maximize the meaningfulness of data leading to personalized medicine. And this technique is very robust and highly reproducible. All the steps along the pipeline really count. So I would put 3 stars on this point. And 2D and 3D multicellular spheroid models that we have done the study with are supposed to be employed with some certain specific purposes, and scientists should be aware of limitation of each cell type. Last but not least, metabolome-specific constructs and fitness level may shed light to more precise and personalized decision-making of cell construction for each patient, hence saving a large amount of unnecessary costs, hopefully. And last but not least, I would like to thank all of the members at the Computational and System Medicine Research Group, our collaborators at George Washington University, Professor Paul Bailey, and the other one at the Lao TPHI, Dr. Shampu Sayason, and my colleagues, Paramay Ganrit and Atapon Titapan, and also our collaborators at Jutarop University for all of the initiatives and experiments so far. And I would like to thank all of the funders, Cholangio Customer Foundation of the United States, NRCT, PMUB, and TSRI. Thank you very much for your attention.
Thank you for a wonderful presentation. Now it's time for a discussion. Any question, suggestion, or comment for Dr. Jutarop?
Peach, thank you so much, and I'm looking forward to your results. And actually, you can present at Chiang Mai. We're planning the next meeting at Chiang Mai University.
Would that be
Will
okay?
you be done by November?
Maybe by the end of this month.
Oh, that's exciting.
If everything goes as planned.
Yeah, because I think at Chiang Mai they have Janipon have mass spec.
Yeah.
So we can set up this
Yeah.
at Chiang Mai as
Yeah,
another site
definitely.
as well.
Yeah.
Like one of the paper you mentioned, they say immunogenetics, metabolomics. What does it mean?
So basically, they look at the immune cells, the metabolism of the immune cells. It's just a term to describe, when we specifically look into the metabolic status of the immune cells. Yeah. Thank you.
Any more questions?
Can I ask about the metabolic profile that you said that metabolic profile can represent the cell type, right? So can we predict that, let's say that cell sample A that have some kind of A metabolic profile, we can transfer that to apply the chemotherapy, or maybe we can predict that the treatment of that CCA in the future, or we already can do that right now?
Well, we haven't gone to that far, but it's one of the aims, for researchers in this field, to be honest. So that is a very good question. So we hopefully be able to characterize such a certain metabolic signatures of different cancer cell lines or cancer cells, even the cancer cells, But one of the main challenge is how could we sort the cells into the same phase, and how could we perform the single cell metabolomics? So that's the major challenge, because like even all the cells have been sorted to the same phase, but it's not that all the cells behave the same way, right? So that means we have to have a good enough technology in order to to visualize the behavioral alteration of the cell at each state and each cell as well. So single cell approach for metabolomic, I would say, is the most challenging amongst all the omics studies. Yeah. But it's the next step ahead. And our center is now developing a single cell metabolomics as well.
Any more questions? If no, Ajahn Phethai.
I think that this is not my field of expertise, so I have to learn more from you. I wonder whether we can use metabolomic in terms of the diagnosis. I mean, if we check the urine metabolomic in the patient, like a CCA, and then can we see the different something, and that can predict or give the diagnosis?
Yeah, that is a very good point. So we can use metabolomics for biomarker discovery in any condition in medicine. So for CCA as an example, we can use metabolomics to discover the metabolite panel that can show the risk population, for example, like periodontal fibrosis. So I have another study running in parallel, so of which I actually finished all the analysis, but I just don't have time to sit down and write up the manuscript. So we use bile acid profiling approach in order to distinguish the periodontal fibrosis, or the PDF, which is considered the risk population, in comparison to the normal population, normal bile duct population, and the cholangiocarcinoma patients. So we managed to come up with 10 bile acid profile that have such a very distinctive and certain, concentrations, to differentiate the PDF from the CCA and normal bile duct. So this is one of the examples. And actually one of the studies conducted by Dr. Mahidol here. So she used metabolomics to characterize the the response patterns of cisplatin, and also you will listen to one of my MSc students talk later on as well, Mr. Natawut here. So he will talk about how we use metabolomics to characterize the responses of these cells as well. So that means metabolomics can be applied into any stages of the disease progression. But the most important thing is how could we design, the good enough study in order to get the good enough data, in order to get the good enough results. So that's the key thing, really. Yeah. And also, as a plus, archive samples are highly not recommended for any omics data. So please bear this in mind. Because one of the first studies after I came back from my PhD, and I was asked to run, genomics and metabolomics analysis, those samples were kept for more than 5 years. And I knew that by heart that it wouldn't work. It's going to be a waste of money. And I told the PI that do not run this, just collect the new samples. And then they said, no, run this. And I said, no, I don't run this. Yeah, it's just a controversy for this kind of, perspective. But yeah, that's my recommendation, that don't use any archive samples for this.
Okay. Thank you, Dr. Jutarop.
Thank you very much.